Good morning or good afternoon, everyone. I'm Julia Caro, Managing Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is The Landscape of Alternative Polyadenylation in the Lung Cancer Transcriptome. The sponsor of this webinar is Lexogen. Our presenter today is Dr. Adriana Zingone, a research biologist at the National Cancer Institute Center for Cancer Research. You may type in a question at any time during the webinar. You can do this through the control panel, which normally appears on the right side of your screen. Just click on the Q&A box on the upper right side of the control panel, and when you click on Send To, please select All Panelists. We will ask Dr. Zingone your questions after her presentation has concluded. So with that, let me hand it over to Dr. Zingone. Please go ahead.
Hi, thank you for the introduction and welcome to everyone attending. Today are going to present some results from one of our studies focused on the alternative polyadenylation in lung cancer. Lung cancer today remain the leading cause of cancer-related death in the U.S. Among different ethnic groups, the African American have the highest lung cancer incidence and mortality. In our lab, we study lung cancer biology and disparity with the goal to identify high-risk patients. For this reason, in addition to conventional genomic and transcriptomic study, we decided to investigate the extent of alternative polyadenylation in lung cancer. Recently, alternative polyadenylation has emerged as a new and independent post-transcriptional mechanism of gene regulation. But what exactly is alternative polyadenylation? Is the selection of an alternate poly-A site on the pre-messenger RNA, leading to the generation of isoform of various lengths, depending on the location of the selected sites. Normally, the signal for the beginning of the poly-A tail, it's located in the distal region of the 3' UTR, but almost 70% of human genes have additional poly-A sites, mostly close to the last exon, but sometimes within an exon of intron. And there are several biological processes normally regulated by alternative polyadenylation. For example, during development, the cell use more distal sites and produce longer transcripts, while in contrast, cells with the high proliferative rate use more the proximal sites, generating shorter isoforms. The deregulation of this process has been associated with various human diseases. The most well-known are alpha and beta thalassemia and also immune disorder, and recently also to cancer. But how alternative polyadenylation is related to cancer? So the 3' UTR, we know that contain important sequence elements, including microRNA binding sites, also motifs for RNA binding protein that regulate the stability of the transcript and also the export from the nucleus to the cytoplasm. As been shown, the normal differentiated cell the distal side is mostly used. So the transcripts retain all of these elements. Cancer cells instead preferentially use the proximal site, generated the short isoform with loss of microRNA repression, increase stability, increase protein production, they lead to an uncontrolled translation, and if this occurs in an oncogene, it can induce cell proliferation and cell transformation. So alternative polyadenylation can activate oncogene in the absence of any mutation. It also can inactivate tumor suppressor gene through the competing RNA mechanism. So recent study shown that a global shortening of 3' UTR is a characteristic of cancer transcriptome. So we hypothesize that lung cancer transcriptome also undergoes to global shortening of 3' UTR. We previously observed that tumor from European American are enriched in cell proliferation and cell cycle pathway. So based on this observation, we also hypothesized an enrichment of alternative polyadenylation in European American. As alternative polyadenylation, it can be regulated by environmental exposure. We want also to determine the contribution of smoking to APA. So the aims of our study are to characterize alternative polyadenylation in European American and African American, to explore also in the mechanism underlying APA, and also to verify findings by functional study. As a strategy, we first selected 100 tumor and 100 non-involved tissue derived from 50 European American and 50 African American patients from our University of Maryland case control study. And then we applied two approaches. One approach is at the sequence level. The Quantification is a novel method for 3' UTR-Seq. Specifically, this method uses an oligo-DT that capture the messenger RNA and also contain the read-one linker sequence at the 5' end. So at the sequencing step, a customizer sequencing prime that are nil to this linker sequence, it's required so that the read starts directly at the 3' end going in a 3' to 5' direction. The second approach we use is at the analysis level. The Express RNA analysis pipeline, it's a new platform that has been developed by our collaborator Gregor Roth with a bioinformatician at the University of Zurich. In particular, the Express RNA APA module, it's specifically designed to study alternative polyadenylation. Can identify poly-A sites that are statistically differentially regulated between two sets of samples. In our cases, tumor versus normal. We are in fact interested in studying the functional status of poly-A sites in lung cancer. But In addition, in order to evaluate the technical performance of our experiment, we introduced in our sample the ERCC spiking control. This is a mixture of 92 polyadenylate oligonucleotides that with different lengths are meant to resemble human transcript. So they function as an internal control. They are present in two different mix, in which the ratio of this oligo is different. So we added mix 1 to the RNA derived from the normal tissue, and mix 2 to the RNA derived from the tumor tissue. From the analysis of this specific transcript, we can have the dynamic range of the read, the fold change response, the dose response. So basically, the analysis is going to tell us how good it's the detection of gene differential expressed in our sample. The dynamic range plot indicates a linear distribution of the read in both samples. The area under the curve, a good level of detection for gene differential express. For example, we had over 90% power in identifying gene with a 4-fold change or 2-fold change. We didn't see any kind of problem in the detection was good, so we moved to the analysis, and the first step in our analysis is the generation of our polyaside database. By using STAR, the 3' UTR sequence data were first processed and mapped to the genome. On average, 74% of the read for each sample align to the human reference genome. Because the oligo-DT we know can cause internal priming events, in order to eliminate this artifact, we use several stringent criteria. We only consider loci that had any of the 18 polyaside sequence in 30 nucleotide window relative to the cleavage site. We also exclude all the positions that have more than 6 consecutive A or more than 8 sparse A in a window of -10 nucleotide upstream and downstream the cleavage sites. Also, we only keep polyasides that were present across at least 25% of our sample. Doing that, we obtain over 122,000 polyasides. We validate our database by comparison to other available databases. Approximately 80% and 90% of our sites overlap with the major used database. In addition, though, in order to avoid poorly used sites, we also filter out the sites with less than 10 read count in either the normal or the tumor tissue. Also, we discard the polyasides. They have less than 5% of read compared to the major polyasides in the same gene. So at the end, we obtain over 37,000 polyasides annotated to 17,000 genes. When we look at the distribution of these sites across the gene, we found 8,000 genes having more than one site. So we focus our analysis to these 8,000 genes that can undergo to alternative polyadenylation, and we apply our DAC-SIC2 analysis to identify regulated sites in tumor versus normal. When we use in particular the adjusted p-value to define poly sites significantly regulated. In case more than 2 sites were identified in a gene, we use only the 2 sites most significantly changed. Also, to be regulated, there had to be also a complete switch in use of the 2 sites. So, if the proximal site was enhanced, the distal site should be repressed. And when we look at the distribution of these regulated sites within the gene, we find a higher number of proximal sites in intronic and exonic region in tumor compared to normal. Our analysis detected 46% of this gene undergoing to alternative polyadenylation. In particular, 8% of them showing a shift in usage of proximal side, while only 12% a shift in usage of distal side, indicating a global shortening of 3' UTR in lung cancer. And not surprising, our pathway analysis of all of these genes with the significant shift in use of proximal sites indicate an enrichment with the pathway commonly dysregulated in cancer. Like as you can see, the ARC-MAP kinase pathway, the NF-kappa-B pathway, the EIF2 pathway. This is consistent with the knowledge the short isoforms are associated with the proliferation. We also look at the gene with the increased usage of distal sites, found to be enriched with pathway normally present in cell undergoing to differentiation or with a very low proliferative status. Also this is consistent with the knowledge that long isoform negatively correlated with proliferation. We then analyze tumor derived from European American and tumor deriving from African American. We found that alternative polyadenylation is twofold higher in European American compared with the other group. In fact, how you can see here, in African American, almost 2,300 genes show a shift in use of proximal side, while in European American, the number is 1,300. Also, we noticed a higher number of intronic and exonic sites used in European American. In our pathway analysis give us some interesting results. We noticed that some pathway associated with short isoform in European American are instead associated with long isoform in African American, like for example P10, also IGF-1. So we found a series of different opposite usage of site. also to determine the contribution of smoking to alternative polyadenylation, we stratified are patients based on the smoking status. So we divide the current smoker and former smoker, and we found that only 10% of the APA events may be smoking related. So our data suggests that smoking is not a major driver of alternative polyadenylation in lung cancer. The pathway analysis of this gene indicating enrichment of pathway previously associated with smoking. But the key question is what drives alternative polyadenylation in cancer? The tri-prime machinery it's very complex and involves cis X elements present at the RNA level, including the polyasite sequence itself, and also a specific sequence present upstream and downstream the polyasite. But there are also many factors that acting in trans. A previous study showed that APA in cancer it's not due to aberration of mutation in this cis sequence elements, but it's more likely due to dysregulation the expression of this factor. So we look at the expression of gene encoding for this factor, and we found that the majority of them to be upregulated in tumor versus normal. And when we extend our analysis to European American, African American, while we found that many of them were equally dysregulated in both groups, we noticed some difference, in particular in gene encoding for the polyA polymerase. The polyA polymerase is the enzyme that catalyzes the synthesis of the polyA tail. It can be present in two forms, canonical and non-canonical. So we noticed that the canonical one, here indicated as a PAP-OLA, is upregulated in both groups. But some of the non-canonical forms, like the ZCCH11 and polyA polymerase D5 and D7, was instead upregulated only in African American. In addition, symplecin, which is a shuffling protein that recruits canonical polyA sites, so is dysregulated only in European American. So our data suggests an enrichment in African American of complex including non-canonical polyA polymerase. And the difference in this non-canonical polyA polymerase is that they tend to target small RNA. But another level of complexity in the regulation of the tri-prime processing is the interaction of the RNA binding protein to the pre-messenger RNA. The RNA binding protein are very difficult to study because they tend to recognize very short RNA motifs, have also multiple domains, and they tend to interact each other. So it's very difficult to identify their recognition binding sites. So we decided to use the RNA motif 2. There is a part, it's a software part of the Express RNA package, and it's specifically designed to identify cluster of binding motif surround the poly-A site, instead to look at very small motif. So when we plot the results of this analysis, here you see just one representative plot. We found an enrichment upstream of regulated proximal site of UC-rich cluster. The predicted RNA binding protein were, for the majority of them, very similar between African American and European American, but in the African American group, we found an enrichment of protein like RF2, ZC3H10, that are also involved in the biogenesis of small RNA. And finally, then we want to test if the alternative polyadenylation was clinically relevant for lung cancer. We identified, so we correlate the gene with the APA switch event with our survival data. We were able to identify a set of almost 20 genes that can stratify our patients in high risk and low risk. Interestingly, this gene set include some non-coding gene like link 00968, and this non-coding RNA has been previously associated in lung cancer as an oncogenic effect in lung cancer. So, in summary, the 3' UTR of transcript, it's globally shortened in lung cancer. African American have a low number of genes undergoing to APA. This difference of on the rate of alternative polyadenylation between two groups, it may be due to some difference in the 3' end processing machinery. We show that smoking it's not a major drive of alternative polyadenylation in lung cancer. APA events are clinically relevant process in lung cancer. With this, I want to thank first my mentor, Brid Ryan, and all my co-workers. Special thanks to Sanju, who did all the survival analysis, all our branch. A very big thanks to our collaborator Gregor Roth, who did all the 3' UTR sequence data analysis. Also, we'd like to thank Lexogen, and in particular Michael Ante, who did also some of the analysis. Thank you, the audience, for listening. And with this, I give a pass to Julia.
Thank you, Adriana. Thanks for that nice presentation. As a quick reminder to our webinar participants, if you have a question, please type it into the Q&A box. And just before we start with the Q&A, we would like to ask our attendees to take a very brief moment after the webinar has ended to take our exit survey and provide us with your feedback. So let's go to the questions right now. What do you think, Ariana, is causing the dysregulation of APA factors?
Sorry, the dysregulation?
Yes, the question is about what is causing the dysregulation of the APA factors.
Oh, yes, the dysregulation of the machinery, it's, so we know, and many previous studies found that this, there is an upregulation of this, but nobody, the mechanism how this goes upregulated, it's not well understood. We know only that these factors are upregulated, and then when the concentration of this factor is increased, there is the use of proximal side it's facilitated, but the how this upregulation occurs, it's not well known yet.
Okay, thanks. And why did you select the only the two most abundant poly-A sites for additional analysis?
Yes, because we want to see the sites basically that are, so we are interested in study any shift in usage of the proximal distal side. We did this based on the sites that have the higher number of the read, the one that the analysis indicate that have a significant shift, and also we did this based also on the sequence of the poly-A site, as I said, based on the specific 18 hexamer sequence. And also the characteristic of the poly-A site. As I said, the sequence of the poly-A site, this upstream and downstream sequence, they define the strength of the poly-A site. So at the end, we select these two major poly-A sites for each gene.
Okay, great. There's a related question to that, and that is, what if the two most abundant sites are actually different in tumor samples and in normal samples? How would you then do the comparison? Or has that not happened? Or?
Yeah, I couldn't hear very well. Can you repeat again?
Yeah, the question was, so you select the two most abundant poly-A sites, but what if they are different in the tumor and normal samples? The two most abundant ones are different.
Yes. As I said, you know, we keep the one have the 18 different sequence, are the one the use was different between tumor and normal. So we took basically based on the characteristic of the poly-A site, the feature of the site, as I did.
Okay. And is there any evidence that APA plays a role in other diseases besides cancer?
Yes, so actually recently it has been shown that a SNP in the gene encoding from BAF create a short transcript. This results in an increased expression of the BAF protein, and it has been linked to an increased risk of autoimmune disease as lupus erythematosus, multiple sclerosis. As well, we know alpha beta thalassemia, in which mutation in a poly-A site, in the canonical poly-A site, lead to the use of more distal side, so generate an elongated transcript that results in a reduced protein production, so a reduction of beta globin protein.
Okay, thanks. There's a question about whether polyadenylation can be used as a drug target, I guess, to develop drug candidates? And if so, which cancer would be a good study model for polyadenylation for that purpose?
Okay, thank you. This is a very good question. There have been already some study, a preliminary study. Basically, the goal will be to target one of these factors that are involved, in polyadenylation. In fact, in lung cancer in particular, the cleavage polyadenylation stimulating factor 4. There has been a study in which has been downregulated to try to target basically the poly-A machinery. At the moment, these are the study, but definitely it's something to consider for as a therapeutic target.
Great, thank you. The difference in alternative polyadenylation between European Americans and African Americans, could that be caused by different tumor histology, and did you test for that?
Thank you for this question, a very good question. So actually, we did that. We looked in adenocarcinoma, patients with the adenocarcinoma, patients with the squamous lung cancer, and we basically didn't find any. We obtained the same result. So the difference that we detect in European America and African America, it's not due to the histology of the tumor.
Okay, thank you. A product-specific question, is it possible to analyze polyadenylation using the Quantification FWD kit?
We use in particular the REV because it specifically allowed the sequence to start from the tripram of the transcript, so we didn't test other one because this one is really specific for this type of sequence. So it worked very well in our end.
Okay, thanks. And then finally, so you mentioned during your presentation that APA results in loss of microRNA-mediated repression. Did you also look for loss of microRNA in the samples?
Oh, yes. Very good question. Yes, actually, we just finished the analysis of MIR loss. We use the gene with the strongest APA switch, and we found in average a loss of 9 MIR for gene, with the range that goes from 1 to 54 MIR loss. And in some of them, we found like MIR 124 or MIR 181 or like 7 that are well known to be involved in lung cancer.
Okay, thank you. It looks like these are all the questions we have. So let's wrap it up now. So let me thank our speaker again, Adriana Zingone, and our sponsor, Lexogen. And as a quick reminder again, please look out for our pop-up survey after you log out and provide us with your feedback. And finally, if you missed any part of this webinar or you would like to listen to it again, we will send you a link to an archived version by email. Thank you very much again for joining us for this genome webinar.
Thank you.